We also looked at the gene expression profile of these children, and we tried to see whether there are any study differences between children. And Upper East, and what we did is they hadn't reported to hospital, but some of them are medication in the previous 2 weeks. Then we did not include you into the study. But those who hadn't taken medication and they were in the community, we recruited them for sampling. We also went to nearby hospitals and we recruited children who are reporting to hospital and who tested malaria positive. And we ended up with 224 samples, 90 subclinical cases, and 134 clinical cases, which we recruited. During recruitment, we did a questionnaire and we took information about their age, whether they had fever, the symptoms, as well as some of these factors. And we tried to determine, we found some variables that were significant, really associated with clinical malaria infection, but we did something further infections. And what you see here is that we show in the dominance analysis, whereby you can see that fever, headache, and parasite density dominate in children who have clinical malaria. By some of these variables, we can actually see that stand out as the most dominant kind of variables, that parasite density 0 to almost 80 years. And you can actually see that parasite density is regardless of the age. We did cell phenotyping and intracellular cytokine staining, and we used flow cytometer to be able to analyze this data. So we stained about 15 different parameters using flow cytometry, and what we found was that of all the 15 different parameters that we stained, T cells with CD45RA positive T cells were significantly higher in clinical malaria infections compared to the subclinical infections. And this could be naive T cells or terminally differentiated T cells. And we also saw that monocytes, these are CD14 positive cells, were significantly different in clinical, or significantly higher in clinical malaria children compared to the subclinical infections. We went further to do the intracellular cytokine staining, whereby we stained some of the cells with the cell markers and as well as some intracellular cytokines. And what we saw was that in monocytes, the CD14 monocytes, they tend to have higher levels of TNF-alpha in clinical malaria children when you compare them to subclinical. And TNF-alpha has been shown for a long time in malaria studies to be significantly higher in children with severe malaria infections. And we also had a similar thing for T cells, CD4 T cells, having relatively higher TNF-alpha levels. So from our analysis, what we had seen was that parasite density, fever, and headache were significantly associated with clinical malaria infection. So what we decided to do was to take children whereby there was no difference between their parasite densities, there was no much difference between their symptoms, and we took 6 children for subclinical and the subclinical, and these children, there was no significant difference for symptoms or parasite density. And we went ahead to do a technique called sequel for single-cell RNA-seq, and this technique is whereby you take the PBMCs and you layer them on a slide which has microwells, and you stain the beads, and you end up with a sequencing library, and you do sequencing. So after sequencing, we analyzed the data using DropSeq workflow, and we further did most of the analysis using our Surat package. And we further did cell-to-cell interactions using cell charts in R as well. So in terms of the bioinformatic analysis that we did a bit of QC, we did clustering of these cell types, we did cell annotation, and we did differentially expressed pathways as well as cell-to-cell interactions. So what I'm showing here is what we call a UMAP. and each dot here represents a distinct cell type. And what we did is PBMC cells for both our cell groups. And what we did is we tried to manually annotate these cell types using already known markers for the different cells. For example, we know CD14 is a marker for monocytes. So what we did was to try and use those different markers to be able to identify all these different cell types, and we label them. But to be able to further identify even the small cell types, because not most of the cell types we have markers for them. What we did was we used the reference defined cell types. This is a CiteSeq study reference which used 162,000 PBMCs, and they used almost 228 antibodies to be able to annotate the different cell types. So we used that to be able to find all the different cell types in our cell population, and we were able to find all the different, for example, B cells. Here we are showing naive B cells, intermediate B cells, memory B cells, and it goes the same for all the different cell T cells, NK cells, as well as the CD14, CD16, and dendritic cells. So when we try to do analysis of all these different cell population, what I'm showing here is the percentage of cells in all the cell types, as well as the different clinical groups. And you can see that the B cells were significantly higher in subclinical infections, but we see that the CD4 cells and the monocytes are significantly higher in the clinical malaria group. And if we look at all the different minor cell subsets, is that we see a similar trend whereby the intermediate B cells are significantly higher in subclinical malaria infections, and the naive B cells as well are significantly higher in subclinical infections. But then when you look at the monocytes, both the CD14 and the CD16 monocytes are significantly higher in clinical malaria infections. And you can see that this is similar to what we had found with flow cytometry, where we showed that monocytes were actually significantly higher in clinical malaria infections compared to the subclinical infections. We did differentially expressed genes, and this is an abstract type of volcano plot whereby we are showing all the upregulated genes in the clinical malaria infections and the downregulated genes in the clinical malaria infections. And you can see we have, just by glancing through it, what we are showing is that we have a lot of immune response genes that were significantly higher in clinical malaria group, because we are showing an adjusted p-value here. We went further to evaluate whether these genes that were significantly higher were actually associated with any pathways, and here I'm showing the different cell types on the different bars across, and here are different pathways, and we are showing the enrichment score, normalized enrichment score for each of these pathways and whether it was significant. And we can see that despite all the other pathways, we can see that several immune response pathways were significant. This is including the IL-2 STAT pathway, the complement pathway, inflammatory response pathway, p53 pathway, and several interferon response pathways, and TNF-alpha plus NF-kappa B signaling pathway. And this was important because we had already shown using flow cytometry that TNF-alpha was significantly upregulated in clinical malaria infections. So we looked at the module score of the interferon responses, and what I'm showing here is the module score for a set of almost all the interferon response genes that we were able to get in our data set. And I'm showing that you can see that the p-value for for example, the monocytes is significantly different, and if you look at the module score, generally is significantly higher for clinical malaria infections compared to the subclinical infections. And this made us to conclude that, monocytes, they actually play a significant role in the expression of interferon-stimulated genes during clinical malaria infection. And when you look at the NF-kappa B target genes, these are the downstream target genes of the NF-kappa B pathway. And you can actually see that the monocytes tend to have higher average expression as well as a higher percentage of cells expressing those particular genes in this subset of genes that we looked at. So moving forward, we looked at the cell-to-cell interactions, and what you can see is that in clinical malaria infections, all the different immune cell types that we found, they tend to have more cell-to-cell interactions in clinical malaria and compared to when you look at them in subclinical malaria infections. And if we try to quantify these interactions, you can see that, so this plot is showing all the different cell ligands or cells that are producing ligands and their respective cell receptors. And here I'm showing the total number of interactions for the receptor and the total number of interactions for the different ligands. And here the color is significant of the total number of interactions between each pairs. And you can see that in clinical malaria infection, generally, if you look at it, there is more cell-to-cell interactions during clinical malaria infection compared to subclinical malaria infection. And when we try to evaluate what is driving these interactions, we found that mostly is the MHC class 1 pathway that has a lot more of the interactions with the different cell types compared to the subclinical malaria infection and MHC class 2 signaling pathway. So I would just like to conclude that what we've seen in this study is that monocytes, CD4 T cells, they play a significant role during clinical malaria infection, and they had higher levels of TNF-alpha. We identified different cell types with significant changes during clinical malaria phenotype. This included T cells, B cells, monocytes, NK cells, and some of their minor cell types, cell populations. And there was significantly higher fractional abundance of monocytes in clinical malaria. And several cytokine genes were expressed during a clinical malaria infection. And these include the interferon and TNF-alpha signaling pathway genes. So this is one of the initial malaria immune single-cell RNA dataset for clinical and subclinical malaria infection. And what we hope to do towards the future is that since we did a cross-sectional study, we hope to now do a longitudinal study where we study children across the spectrum so that we are able to understand how these malaria infections occur at the cellular level and their gene expression patterns. And ultimately, we will be able to contribute to the development of a malaria disease atlas, and we hope to not just stop at the blood and describing the immune cells, but also go ahead to look at the bone marrow and liver as well. I would like to acknowledge my supervisors for my PhD. I would like to acknowledge the Department of Biochemistry, Cell and Molecular Biology at the University of Ghana. I would like to acknowledge Dr. Bediako, Dr. Aniwe, Nancy, Joyce, and Dr. Abraham, where I did my work in Avrongo. And I would also like to acknowledge Auto Lab in Glasgow. And finally, the Deltas Africa grant. I was one of the awardees for the Deltas 1 grant, and I would like to thank the Science for Africa. I also like to thank the Chalet Club, who were instrumental in providing the single-cell RNA-seq work. Thank you very much.
Thank you. Thank you, Collins, for the interesting presentation. It's time for questions. Okay, we have 1, 2. Any more? Okay, so yeah. Thank you.
I missed it, but did you selected these on the basis that their parasitemias were the same?
Yes.
Okay.
Yes.
Have you ever done a comparison, just out of interest, between low and between parasitemias in the 12?
Yes.
In this?
So we were cognizant of that, and that's why we tried to select individuals where there was at least no much for both of the groups was less than 15,000 parasites per microliter, and we ensure that they had all the clinical symptoms. There was no much significant difference, and this was just to try and adjust for all the other parameters that affect malaria and the
data. While you were presenting that, we did the entire expressed transcriptome in children with non-SMA versus SMA.
Yeah.
And those children with severe malarial anemia have very similar profiles, though. When we did deconvolution analysis with CyberSort X, which is a way to essentially just go in and characterize cell types, the largest signal we got was actually neutrophil activation. So there's a higher level of neutrophils in non-severe disease, but then when you looked at neutrophil activation with gene ontology, those particular clusters of those neutrophil responses seem to be pretty important, at least in severe malarial anemia. And so I didn't see neutrophil responses in there, but I probably missed it because I'm jet lagged. What did the neutrophil responses look like?
Yeah, so we took PBMCs and we stored them in liquid nitrogen before we used them.
That's why I didn't
So
see it.
Usually,
Thank you.
neutrophils.
And then just a really
yes.
quick comment / follow-up question, and not a criticism. How do you know that the children were not treated?
Are they community cases or at the hospital? So we just asked the question whether you've taken any medication for the past 2 weeks.
I know this won't surprise you, but humans lie, and particularly and particularly when it comes to medicating their children. So I don't think it impacted on your analyses, but
Yes.
if you were to get a criticism, you can simply look with TLC that's really quick and easy in urine to see if there are any antimalarials on board. Just a thought. Thanks.
Okay. All right.
Okay, thank you very much, Collins. Thank you.
Thank you very much.
Okay, our next presenter is Cynthia Amisigo, and she's going to talk about acyl galates disrupt trypanosoma brucei lipid droplets. Okay, you have 10 minutes. Oh, 12. Okay.
Okay, good afternoon. I'm Cynthia Misgo, and my talk is on alkyl galate disrupt trypanosoma brucei lipid droplets. So trypanosoma brucei is a causative agent for African trypanosomiasis, and based on its mammalian host, the disease can present in two forms. We have the prevalent, and with an estimated loss with about $4.5 billion annually in livestock production. Now, the life cycle of trypanosome shuttles between the insect vector and the mammalian host, where in the mammals, the parasites are found in the intravascular and extravascular compartments of the host. And recently, these parasites have been shown to be present in the adipose as well as the skin. And with this, during the life cycle of trypanosomes, they encounter different materials in terms of their environment. And one way that they respond to these different environments is by changing their metabolism. And in the adipose tissues undergo genetic rewriting, where they increase the gene expression levels of lipid metabolic genes in order to survive in that environment. Also, due to the prevalence or the presence of nutrients, they're able to change this lipid metabolism to either survive or to generate energy. So this kind of hammers the importance of lipid metabolism to the survival of the parasite. So one key component or important organelle in lipid metabolism are the lipid droplets, and these are fat-storing organelles that originate from the endoplasmic reticulum and are found in the cytosol. So they are made up of a phospholipid monolayer and also contain neutral lipids, including triacylglyceride and sterol esters. Now, these organelles are important for cellular processes, including lipid metabolism and also energy metabolism. Now, using the tri-chip database, we identified 12 lipid droplet proteins, and for this presentation, we are focusing on the lipid droplet kinase, because studies have shown that this is the only kinase that have been shown to be located on the periphery of the droplet and has been shown to be important for both the maintenance and synthesis of the lipid droplet. And here we have the presence of the lipid droplet shown in the yellow, and on the periphery we have the lipid droplet kinase shown in the red. Now, this study suggested that a downregulation of this lipid droplet kinase caused a loss in the lipid droplets as well as a reduction in the number of lipid droplets, also emphasizing the importance of this kinase to the maintenance and synthesis of the lipid droplet. So in our quest to understand lipid metabolism in trypanosomes, we decided to look at compounds that could play a role or interfere with the lipid metabolic process, and we narrowed down on a set of compounds known as the lipophilic alkyl galates. So these are esters of gallic acid that contain a hydrophilic bioactive molecule and a lipophilic domain. And studies have shown that some of these alkyl galates possess good anti-trypanosomal activity. So based on the structure and based on the lipophilicity of these compounds, in addition to their anti-trypanosomal activity, we hypothesized that these compounds could play a role in bloodstream forms of trypanosomal brucei. So to begin with, a set of alkyl galates were chosen, and these were based on their varying alkyl length chain as well as their lipophilicity. In addition, a standard trypanosome drug, that is diimidazine acetate, of these alkyl galates and using the Lama blue cell viability assay. And here the data shows that these alkyl galates possess good anti-trypanosomal activity with IC50 ranging from as low as 0.04 to 4.7 micromolar. And because these galates have been shown to be very active as compared to downstream assays. So to begin with, we decided to look at the effects of this galate on the lipid droplet kinase. So what happened here was that we tagged lipid droplet kinase protein at the N-terminal using image. So this imaging will help us detect the presence of these kinase foci in the cells. So this is a representative cells as well as the DMSO control. We observe that there's a presence of the kinase foci dotted in the cytoplasm of the cell. And comparably, looking at the cells that were treated with the galates, we observed that most of the cells appear to have lost this kinase foci. And this was in contrast when we compared to the standard drug, where most of the cells still maintain their foci. So that means that the disruption or loss into this kinase foci is specific to the activity of the galates. So this is a larger picture for you to appreciate galate. You see that most of the cells lose this kinase foci. So in terms of quantification, we look at the percentage of cells that have this kinase foci within the different treatments. And here it was observed that oral galate also lost this kinase foci. So we wanted to know at what point do these cells start actually losing their kinase foci? And it was observed that as early as 30 minutes, about 50% of cells that were treated with octal galate showed a significant reduction in the percentage of cells that lost the kinase foci. And there was a gradual loss from 30 minutes all the way to 6 hours. However, for the cells that were treated with oral galate, a significant loss in the kinase foci was shown when the cells were treated for 2 hours, from which they gradually declined to 6 hours. So this indicates a rapid loss in the kinase foci, and that could be suggestive of the mode of action of these compounds.